Hello. So we're continuing with um, life science and cell biology in a nutshell. So we're learning about we are learning about the endoplasmic reticulum. We saw what it looks like. It has cisternae or folds. So they're flattened sacs which hold the ribosomes, which are used for protein production. Here you see a micrograph with the ribosomes, which are the darker spots on the folds. The smooth endoplasmic reticulum doesn't have ribosomes, but it does have folds and is contiguous with the rough ER. So this is devoted for production of lipids and steroid hormones and so fats are production and metabolism of fats and steroid hormones. It is useful also for detoxification of organic chemicals and converting them to water soluble products. So then and sarcoplasmic reticulum is used for storing ions that are helpful, such as calcium, that are needed for cell functions. So the endoplasmic reticulum starts at the outer edge of the membrane of the nucleus and goes outward, the rough and the smooth, and etc. You have here staining of the nucleus and the surrounding ER. Here you see this as well. The rough ER, the smooth ER, and what the folds look like. This is the nuclear membrane in the brown, and the ER. So in gray you have vesicles with the vesicles where that have, and then ribosomes as well. So ribosomes are where proteins are produced. So endoplasmic reticulum once again. Here's smooth. This is the rough, which has ribosomes. The endoplasmic reticulum is in cells of eukaryotes. It has an interconnecting network of flattened membrane-enclosed sacs or tubes known as cisternae. So here's 3D showing the nucleus and the ER, which surrounds it. The membranes, it's very beautiful. Here as well, the nucleus and the ER. Very lovely images. Here as well, the smooth and the rough. The rough has ribosomes, the smooth does not. Here is a bacterial cell ER, endoplasmic reticulum, showing the smooth ER, rough ER with membrane-bound ribosomes. So membrane-bound ribosomes mostly create proteins that are bound for excretion from the cell, while free ribosomes usually create proteins which are destined to stay inside and be used by the cell. Mitochondria is the powerhouse, which we'll learn about in a bit. So ER proteins synthesized are destined for export from the cell. Many the hydrolases in lysosomes remain inside the cell. So hydrolase is a type of protein which is synthesized on the ER. Other proteins are anchored in the membrane of internal organelles or in the cell membranes. The proteins that are created. It is presumed that each protein has some type of marker that fits a specific location in the cell. So, proteins synthesized on free ribosomes have segments that bind to specific receptors on the outer membrane of mitochondria, chloroplasts, or peroxisomes, depending on where the proteins are supposed to be. So they're taken up by these organelles as a result of their markers. In the case of proteins synthesized in the ER, the RER, both the hydrolases destined for lysosomes and the secretory proteins are found initially in the same portion of the ER lumen. Studies have shown that these can be distinguished on the basis of their carbohydrate residues. The carbohydrate residues of lysosomal enzymes become modified in this cis-Golgi by the addition of certain phosphate groups. This critical modification allows the enzymes to bind to specific receptors on the membrane of the Golgi, which directs them into vesicles leading to a lysosome rather than a secretory vesicle. In the lysosomes, proton pumps create an acidic environment, causing the release of the lysosomal enzyme from the membrane-bound receptors. Much of this sorting activity is mediated by coated vesicles containing the same fibrous outer protein, clathrin, used in endocytosis. These sorting vesicles also contain associated smaller proteins. So again, showing the ER, the rough and the smooth, cisternal space between cisternae, nuclear pore, nuclear envelope, showing the ER. It's a contiguous membrane, which is so connected to the outer of the two nuclear membranes, forms a membranous network throughout the whole cell, divided into two parts, smooth and rough. Rough has ribosomes attached to a cytoplasmic surface responsible for the synthesis of transmembrane proteins, which are proteins inside membranes, and those that are secreted to the extracellular space. The smooth is involved in lipid and steroid synthesis. The ER is one of the major storage sites for intracellular ions, maintained in order to maintain homeostasis in the cell by regulated release and uptake of ions. Thank you.